Transcription and translation are the two main steps of the process of gene expression. There are 3 stages in transcription: initiation, elongation, and termination.
Transcription occurs in the nucleus.
In initiation, RNA polymerase binds to a promoter on the gene it will be transcribing.
The promoter will direct the RNA polymerase to a location where the enzyme can begin transcribing on the DNA. Next, the RNA polymerase moves along the antisense strand of the DNA in the 3' to 5' direction. This step is called elongation, where the RNA strand will increase in length.
Antisense is the template strand that RNA polymerase will be transcribing.
RNA polymerase adds a complementary RNA nucleotide for each nucleotide along the template strand.
The new RNA strand is almost identical to the non-template or coding strand of the DNA, with the exception of thymine being replaced with uracil.
The final stage is termination. The RNA polymerase will not stop transcribing until it is instructed to do so. The finished RNA transcript is called messenger RNA or mRNA.
mRNA modification processes pre-mRNA so it can be translated into a protein. mRNA modification also occurs in the nucleus.
The next step is RNA splicing. Here, certain parts of the RNA transcript, called introns, which are unneeded parts of the sequence, are removed by a protein called the spliceosome. The remaining sequences, called exons, are glued together by the spliceosome. This process creates the mature mRNA.
A 5' cap, which is a modified guanine nucleotide, is added onto the beginning of the RNA transcript. Then, a poly A tail is added onto the end. The cap and tail protect the transcript and help it leave the nucleus to prepare for translation.
The mRNA travels out of the nucleus into the cytoplasm after transcription is done. Translation begins when a small ribosomal subunit binds to the mRNA.
Next, the initiator tRNA binds to a start codon. After that, the large ribosomal subunit joins to create the translation initiation complex.
tRNAs bind to their codons. The existing amino acid chain links with the incoming amino acid.
The last step of translation is termination. In termination, the finished chain of amino acids is released. A stop codon must enter the ribosome to trigger a reaction that will release the polypeptide chain from its tRNAs, and the chain will be able to leave the ribosome.
This is how we went from DNA to mRNA to a protein.